Hi, good afternoon. We will finish lecture 3 today and probably start a little bit on 4, but we will end a little bit early today because a lot of you still haven't done the quiz, and I want you to have some time to maybe prepare for that, study more, or take the time to complete it before it gets too late. So let's keep going with our lecture 3 materials. Last time we spent quite a bit of time talking about the endomembrane system. Right? It's a network of membranes that are responsible for synthesizing proteins and lipids and packaging the proteins and then modifying them and saving it for later, or you can send them out of the cell. The endomembrane system is also responsible for breaking down macromolecules, detoxifying drugs, and so on and so forth. So today we will take a look at the rest of the cell and see what other organelles are there. Starting with this one, okay, peroxisome. Peroxisome looks very similar to a lysosome. Like, in fact, this is a real picture of it. This is a lysosome, and this is a peroxisome. They are both like, and here's another one. They both look like circular in structure. It's very hard to tell based on just a diagram like this. So it's not something I'm going to ask you to label on the test. All right. There are other methods to identify which one is which, but it's important to remember that the lysosome, which is what we did last time, the lysosome is part of the endomembrane system, and it helps break down molecules as well as worn out organelles. And sometimes it's even able to trigger programmed cell death, right? And here we have the peroxisms. Peroxisms, they carry out a kind of reaction called oxidation reaction. Oxidation reaction. Now, oxidation reaction, to keep it simple, basically involves reaction where electrons are taken away. Right? And these kind of reactions, well, the one that happens in the peroxisome anyways, they release hydrogen peroxide, or H2O2, as a byproduct. Right? And H2O2 is very damaging. You cannot let it like roam free in the cell. It will bind to various things and cause damage to your proteins, to your DNA, and we just cannot allow that. So these kind of reactions, we have to contain it within the peroxisome. Okay, so they help break down fatty acids and amino acids. It's very similar, right? Like, if you think about it, it's very similar to the function of the lysosome, but the kind of reactions that are happening is different. In the lysosome, the reactions are carried out by enzymes. In the peroxisomes, we have a different kind of reactions called oxidation reaction. The peroxisome help detoxify many poisons that enters the body, such as alcohol in the liver. So now, by this point, we have learned 3 different organelles that can help us break down toxins and medications, right? There's the smooth ER, there is the lysosome, and now there is also the peroxisome. So here, it's just a question that's related to the note we took last time. If you track the movement of a protein produced in the rough ER, it can end up in all of the following locations except, okay, so let's just recap quickly. If it's made in the rough ER, you're going to package it in a vesicle, right? And that vesicle is going to move to the Golgi, where the contents get sorted, they get modified and repackaged into a new vesicle. And we wrote down where that vesicle can go last time, right? It can embed itself in the membrane. It can stay as a vesicle in the cytoplasm until it's needed later. It can become the lysosome, right? So this is the exception. Okay? The peroxisome is not even part of the endomembrane system, and so it's not really involved in this at all. Okay, next we're gonna talk a little bit about an important organelle that helps power our cells. Okay? It is called a mitochondria. The singular for mitochondria is mitochondrion. Okay, just spell the whole thing. Okay, so this is singular, and this here is plural, and that is the site of energy production. Okay? Part of what we call cellular respiration takes place in the mitochondria. And if you remember from the second lecture, cellular respiration is this process where you take glucose and you react it with 6 oxygen to produce 6 carbon dioxide, 6 water, and more importantly, energy. Okay? Some cells have only a few mitochondria. Others have more than 1,000, depending on the energy requirement of the cell. Your heart cells needs a continuous supply of energy so it can keep on beating, right? So needless to say, they would have lots of mitochondria. Your fat cells, they are just storing the lipids. They don't really need to, move too much. And so you wouldn't expect to have too many mitochondria in a fat cell. That's what the mitochondria looks like in one of those electron microscope image, and this is like a drawing of it. So because understanding the mitochondrial structure is key in helping you learn metabolism in lecture 5. So I want to take this time to just draw it out with you once so that you're kind of aware of the key features of the mitochondria. So here we go. All right, so the mitochondrion looks like this. It's like a jelly bean, and of course it's 3D, but we're gonna draw it 2D, okay? We're gonna cut it open and see what it really looks like. So there is an outer membrane. Outer membrane. The membrane is the same kind of material that makes up the cell membrane. And by now you should know that would be the phospholipids, right? In addition to the outer membrane, the mitochondrion also has an inner membrane, but this inner membrane is all folded up. It folds up like this. So this is the inner membrane. And like I said, inner membrane folds up into, most textbooks describe it as finger-like projections. They kind of look like short, stubby fingers, I guess. Folds up into finger-like projections called cristae. This helps increase surface area. For chemical reaction. Okay. So the cellular respiration, that equation I wrote for you, is a chemical reaction. And you need to have a lot of area available for those kind of reactions to take place. Generally speaking, the more surface area you have, the faster you can do the reaction. So this is like the membrane, and instead of straight like that, you fold it up like this, right? You fold it up, okay? And then you fold it a couple times so you can pack a lot of things inside the mitochondria. All right, so that's the outer membrane and that's the inner membrane. Now, between the membranes, between the outer and the inner membrane, there is an area. Okay, that area is called the intermembrane. Intermembrane means like it's between the membranes, essentially. Intermembrane space. Each of these things have a direct role in allowing the production of energy. And we will learn all about it in lecture 5. Okay, finally, the innermost portion of the mitochondria, it's called the matrix. It's called the matrix. Okay, fun fact. Mitochondrion contains its own DNA. Okay, that's separate from the 46 chromosome that you have. It has its own DNA. And also, fun fact, the DNA in the mitochondria, all of which are passed on from the mother. Okay, all your mitochondrial DNA comes from the mother. Right? The egg is really big. Sperm is, relatively small. And when the sperm fertilizes the egg, only the DNA of the sperm goes into the egg to fertilize it. It doesn't, none of the organelle from the sperm goes into the egg, right? So the egg containing the mitochondria would be all maternal mitochondrial DNA. And then so in addition to your 46 chromosome, half from mom, half from dad, you will also inherit mitochondrial DNA, which come only from the mom. Okay, next we have chloroplast. Chloroplast. Okay, chloroplasts, they are found in plants, not in animals. Okay, actually, they can be found in other things like protists. Okay, now I know I didn't really talk too much about protists, but example of protists would be things like amoeba and the paramecium I told you about, right? Okay, those belongs to another group of organisms called protists, some of which have chloroplasts. The amoeba does not, the paramecium does not, but other kinds they do. Like kelp, for example, is an example of a protist, and they have chloroplasts in them. Okay, so they are the site of photosynthesis. Now, photosynthesis is the reaction by which plant makes their own food. And so the reaction goes like this. There's carbon dioxide, you need 6 of them, plus 6 water. And in the presence of sunlight, or really any kind of light, it will become food, right? C6H12O6, that becomes the glucose. The glucose can then be stored as starch. We learned that before, and produces oxygen. Right? So this is photosynthesis. And it happens in the chloroplast. Chloroplast contains chlorophyll, which allows the plant to capture sunlight. Chlorophyll is what makes a plant green. Okay. Just like the mitochondria, the chloroplast has 2 membranes. Okay? It's double membrane. Right? We're not gonna learn about the structure of chloroplast. This is like human biology. So here's a picture of it, but you don't have to memorize any of these things. What you should know though is the relationship between the reaction that goes on in the mitochondria versus the reaction that goes on in the chloroplast. Okay. Specifically, you can see in the mitochondria, that's where cellular respiration takes place, and the ingredients, the reactants, are glucose and oxygen. And then you produce carbon dioxide and water. But the product of cellular respiration, they become the ingredients for photosynthesis. And the product of photosynthesis are actually the ingredients for cellular respiration. Okay. So the products of cellular respiration are the reactants for photosynthesis. Okay. And then similarly, you can write it yourself. Similarly, the products of photosynthesis are the reactants for cellular respiration. So for plants, they have both chloroplast and mitochondria. And what they do is they make their own food, and then their mitochondria can burn the glucose. For us, like the source of the glucose would be from our food, right? We have to eat the food, we have to consume it before we can burn it. But the plants, they can just use the sunlight to make them. Okay, so these two reactions, supposedly, it's gonna, form a balance in nature, right? Where animals and plants, they have this equilibrium, but of course, with human activities, we have way more carbon dioxide that are being released in the atmosphere than the plants can capture. So the balance is a little bit tilted. Any questions so far?
I have a question. So how would you explain that in order? Which one comes first?
Oh, that's a great question, right? That's like a chicken and egg question. Like, did the chicken come first or the egg come first, right? It's a cycle. You can think of it as a cycle. There's no beginning, there's no end, right? Just think of the animal and the plant coexisting, and then it's just circling over and over again.
Like, I'm not understanding how to explain it. Like,
Explain what
like
specifically?
this whole process.
Mm-hmm.
Um, like, so basically, is it like the differences between the chloroplast and the mitochondria? That's what we supposed to know?
Okay, so the chloroplast and mitochondria, these are two different organelles, right? And the chloroplast is where the photosynthesis reaction is going to be happening. Okay, so what is photosynthesis? Photosynthesis involves taking the water and the carbon dioxide and then creating glucose and oxygen. That's photosynthesis. You with me so far?
Yes.
Okay, now cellular respiration is like the opposite of photosynthesis, and it takes place in the mitochondria. And that reaction involves taking glucose and oxygen and then producing carbon dioxide and water. So the key takeaway here is that the two processes here complement each other. Okay, if only chloroplasts exist, then you would run out of carbon dioxide and water essentially. Okay, and if only mitochondria exists, then you would run out of glucose and oxygen. But because both of these exist in nature, right, you are able to recycle the carbon dioxide essentially and change it back into glucose. And then you can use the glucose and then recreate the carbon dioxide through the mitochondria. So those two processes counteract each other, they balance each other. Is that a little bit better, or you have a follow-up question?
That's a little bit better. So basically, photosynthesis produces water and carbon dioxide.
Photosynthesis uses carbon dioxide and water,
Mm-hmm.
right,
To
to
produce
produce glucose and oxygen.
And then cellular
Respiration.
respiration produces uh, the carbon dioxide and water
Using glucose and oxygen, right?
using glucose and oxygen. Okay.
Yeah. So this happens in our body, right? And we breathe oxygen, we breathe out carbon dioxide. We eat
Okay.
glucose, and then we produce water, like we pee, for example.
Okay. Okay, I understand.
Good.
So like basically plant breathes in carbon dioxide, right? And
Yeah.
they produce, okay, I get that.
Right. Yeah, that's why they say, right? Like plant more trees to help absorb the carbon from the environment, right? They are essentially a carbon sink. Yeah. Any other questions before we move on? Great. Now, there are other organelles that are found in plants, but not in animal cells. Another example of that is something called central vacuole. Right? This is a picture of a plant cell, and never mind what these other stuff are. The focus here is that there is a big filled up sac here, and that is basically what the central vacuole is. Okay, it takes up more than half of the volume of a cell, of the plant cell, and its main function is to store water and other nutrients. When you water a plant, the root absorbs it, and then they would store all of the water inside the cell, and they will use it slowly. But more importantly, the water is actually going to help the plant stand up. If you have house plants and you forget to water them for like a couple of days, you can see the leaves kind of like become wrinkly, and they kind of wither, right? Shrivel up like that, right? That's a sign that the cells are not having enough water. Once you water them, it fills up the vacuole, and that actually creates pressure to help the leaves open and stand up against gravity, essentially. Some central vacuole in flower petals will have some pigments in it. Pigments are basically color, okay? And some plants may have poisons stored in the central vacuole as well. Okay, so it's like a storage facility, mostly water and other nutrients, but it can specialize and store pigments or poison depending on what kind of plant and which part of the plant you're talking about. So central vacuole is one type of vacuole. There is another kind of vacuole called contractile vacuole. In a contractile vacuole, the function is to continuously pump water out of the cell so it doesn't burst. So this organism here is the paramecium that we talked about previously. It's a pond. It lives in a pond, and it's a type of protist, okay, just like the amoeba. And one of the challenges for single cell organism to be living in a pond is that the water will continuously rush into the cell. And if they don't pump those water out, it's just going to fill up like a balloon, and it pops and die, right? So the contractile vacuole does that for them. Okay? There is the contractile vacuole. It basically squeezes, and then the water goes out, and then it fills up with water again, and then it's squeezed. It must continue to do that all the time. Otherwise, it will definitely die. Okay, so we do not have contractile vacuoles. Plants don't have them. Only these protists might have them sometimes. Another feature that's found in plant cells and not other cells, not in animal cells anyway, are cell wall. Right? So plant cell wall, we first learned about it when we talk about cellulose in lecture 2. And cellulose is this very tough fiber, tough polysaccharide that is made up of repeating units of glucose. And they are able to form a very rigid structure around the cell, okay, around the plant cell. So that's the nucleus, that's the cell membrane. And then now you have this cell wall on the outside, right? And the major function, the main function of the cell wall is to prevent the cell from bursting. Okay, remember, you have the water in the central vacuole, and the central vacuole can get quite big, right? And if you don't protect the cell, it will burst, just like the paramecium would. Okay, there's no pumping mechanism. In fact, the plant cells actually kind of like the water. They need the water to survive, right? To do photosynthesis, so they don't mind storing the water, but you just gotta encapsulate it and make sure that it's not gonna burst open. It's like blowing up a balloon, but then you put the balloon in a cup first, okay? And so it prevents it from popping prematurely. All right, this is a very long question. Why don't you guys give it a try? Okay, that was more than a minute, and unfortunately, I only have 4 responses, but we gotta move on. So I'm gonna end the poll. Thank you for those who chose something. We're looking for the false statement. Smooth endoplasmic reticulum is part of the endomembrane system. That is correct. Okay, the endomembrane system includes nuclear envelope, rough and smooth ER, the Golgi apparatus, as well as the lysosome. Okay, so that's a correct statement. We should not be choosing it. There is an abundance of mitochondria in muscle cell. That is also true. Muscle cells require a lot of energy to contract and to produce movement. So it's not surprising you would need a lot of mitochondria to supply that energy for them. All cells have cell wall to protect itself from the environment. That is not true. We're looking for the false one. That's a false statement. Animal cells, for example, do not have cell wall. Hydrogen peroxides are found in peroxisomes, not lysosome. Okay, we learned that today at the beginning of the lecture. Chlorophylls are found in chloroplasts. That is correct. So C and D are the false statement. Okay, so like I said, the quiz will have some of these multi-select questions where you have to choose like more than one answers. Okay, we're going to spend the last little bit of the lecture talking about the cytoskeleton. So here we go. The cytoskeleton refers to a network of fibers that are found throughout the entire cytoplasm. Okay, some of which actually attach onto the cell membrane. Others, they are just traversing the entire cytoplasm. Okay, they are important for providing support, so the cell is not gonna like collapse on itself. And in some cases, they are important for producing movements for the cell or allowing movement for things inside the cell. Okay, I'll give you specific examples of those in a bit. There are 3 types of fibers that I want to introduce to you related to cytoskeleton. We have what's called the microfilament, microtubule, and intermediate filaments. Okay, so let's take a closer look at each one of these and see what they do. First one, microfilaments. Microfilaments are made up of 2 intertwined actin protein strands. Let me go back to here. This here, that's like one strand, and then this is another strand. Okay? These are proteins, and each of these balls, they are called actin. Okay? Actin. So multiple actins can come together and form this whole microfilament. Now, where do you find the microfilaments? Well, they are actually found throughout the entire cell. This here is a picture of a cell, okay? Like this is the cell membrane. You can't see it, like I just know it's there. You just have to trust me, okay? These are called fluorescent microscopy. You can use fluorescent tags to highlight certain parts of a cell. So in preparing this picture, the person used a green fluorescent to highlight the actin proteins, and then they use a red fluorescent to highlight the DNA in the nucleus. So this red circle here, that's where the nucleus would be. And you can see everything else here, this like all these green fibers, they are all actin proteins. They're all microfilaments, and pretty much they form a web that goes all around the cytoplasm. And the function of that is to allow various organelles to attach onto the actin filaments. Okay, the mitochondria is not like floating around randomly. Okay, the Golgi body, the rough ER, all these things are actually quite organized in a cell. Okay, and the way that they can stay put is by anchoring themselves onto the actin filament. Okay, you can think of the actin filament as one of like a Velcro board or something, and or like what's another way to think about it? Like those cork boards where you can stick like thumb tags on, and then you can hang different things, you can pin different things on it. Okay. Another function of the actin is actually to allow cell movement. Okay. Not all cells are able to move around in the body necessarily, but some do. Okay, some cells can crawl around from one place to another. White blood cells do that, for example. And in order for the cell to move in one direction or the other direction, it involves, so here, allow cell movement, where actin proteins are reorganized in the cytoplasm. Okay, just try. Let me try to explain this to you first. Okay, it's hard for me to show on a, in a virtual environment, but try to follow what I'm trying to say. Okay, let's say the cell wants to move this way. Okay? Then what it has to do, and this is for real, it actually needs to break down all the actin over here, and then they ship it all the way to the other side, and then they build the actin over here. Okay, so they build more actin here. So as time goes on, the left side is basically gonna shrivel up, okay, and then the right side, because we're building more actin, is gonna extend forward, right? So this reorganization of the actin actually push the cells in one direction. Okay? So that is what the microfilament does. It provides attachment sites and it allows cell movements. Next, we have the intermediate filaments. So same staining technique, but this time we use the fluorescent to highlight the intermediate filament instead of the microfilaments. But if you see the location of these things, it pretty much overlaps quite a bit with the actins, right? Okay, so they're kind of everywhere. The intermediate filament is particularly important for connecting cell to cell by forming these intercellular junctions. Okay? So let's say I have a cell here, and then I have another cell here, and we want to staple the cells together. Okay? We don't want to have a gap between them, so things not going to leak between the two cells. The intestinal tract, you need to have these kind of sealing. Otherwise, the food's gonna like leak out, right? And go everywhere. We don't want that. So we want to produce these tight connection, these tight junctions, and we need a little bit of intermediate filaments to do that. Think of them as staples. Okay. They also play a very important role in stabilizing the nuclear envelope. As you know, the nuclear envelope surrounds the nucleus, and it prevents the DNA from leaking out, and it also controls what goes in and comes out of the nucleus, right? So you want the nuclear envelope to be intact and not like falling apart. That's the job of the intermediate filament. There is condition called progeria. Has anybody heard of this before, or you seen a picture similar to this? Does anybody know what's going on here? Anybody want to share?
I've seen a picture similar.
Yeah. Did you know, have you read about it though, or?
No, I've only seen pictures.
Right, okay. So this is called progeria. It is sometimes called rapid aging disease, okay? The individual in this picture is no more than 14 years old by age, okay? And yet he has all the features of an old person, right? And one of the causes of these kind of aging diseases, the progeria particularly, is caused by a mutation in a protein called lemon A, right? And this protein is an example of intermediate filament. So what happens is, with the mutated lemon A, the intermediate filaments are no longer able to sustain the integrity of the nucleus, okay? Because the nuclear envelope is no longer intact, and so it leads to altered chromatin structure, and it interferes with gene expression, all of which adds up to causing the person to age rapidly. Okay, this is a very, very rare genetic conditions, and the thing about rare conditions is, you know, there's not a lot of research done on it, right, to find a cure. People are trying to study whether gene therapy is going to have an effect on, you know, creating a cure, and there are some success in mouse model, okay, where they're able to rescue the mouse from these predisposed genetic conditions. So you can read more about it on your own if you're interested. Last, we have the microtubules, okay? The microtubules provide a long track for vesicle to move through cells. Remember last class, we said the vesicle butts off from the rough ER, and then it needs to go to the Golgi body, right? But like, how does it know where to go, right? Like these things are not really like thinking, right? You know what I mean? Like the cell is alive, but the vesicle is not. So how does the vesicle know where to go, right? It turns out the microtubules, they form these long tracks for the vesicle to be transported along, okay? And there is actually a protein called kinesin, and this is for real, okay? Like they actually do look like this. You can Google it on your own if you don't believe me. They look like a person, like a stick person, okay, without the head. Okay? And then the arms here, that's where they would grab onto the vesicle. And then they would walk along the microtubules and carry the vesicles to the Golgi body, for example. Okay? But not just that. Even after it comes off from the Golgi body, remember, it have different places it can go, right? It can leave the cell, it could go and become part of the cell membrane, it might stay in the cytoplasm for a little bit. So all these places that it needs to go has to be transported along these microtubules, right? So things are not random. If we rely on just chance and randomness for things to happen in a cell, then nothing ever gets done, okay? It's not very efficient. Right, so life has evolved to have these systems in place in the cytoplasm so that things can happen in an orderly manner, which brings back to our original point, right? One of the characteristics of living things is that there are complexity and order associated with it, all the way down to the molecular level. Speaking of microtubules, there are other instances where they're needed. For example, in animal cells, we have a pair of centrioles. Okay, plants don't have these, but we do. Okay, I'll have a chart that's coming up later that tells you, the animal exclusive one and the plant exclusive organelles. Okay, so don't worry about that for now. So we have these centrioles, and the function of the centrioles are to produce these microtubule fibers that help guide the movement of chromosomes. Right? Remember last class I said when a cell divides, they need to pack up all the chromatin, cause them to condense and become the chromosomes, and then you got to move them around, right? Because we have 46 chromosomes, and when a cell divides, we got to make sure that the two new cells each, like you get 46 and you get 46, right? So we have to move the chromosome around, and the way to do that is by creating these special microtubule fibers called spindle fibers. Okay, if you don't remember the name for now, that's fine. We will learn more about the spindle fibers later on when we discuss mitosis. So that's what they look like, okay? On quiz number 2, which is happening, I forgot, like, first week of October or something like that, there will be questions where I give you a picture and I ask you to identify what it is. So make sure you familiarize yourself with these pictures. These kind of remind me of like those, pasta. Sometimes they have these shapes, right? I don't know what you call them exactly, but I've seen them in the supermarket before. The microtubules also form 2 other structures, okay? Cilia and flagella. Okay? So let's talk about cilia first. The singular for cilia is cilium, but we rarely say that, okay? Because if a cell has cilia, it's going to be covering the entire cell body. Okay, so in the case of the, again, paramecium, the pond living thing that has the pumps in them, right? How do they swim around in the water? Well, their body are covered by these cilia, and the beating of the cilia allows them to move, one way or the other. We also have cilia in our body. I told you that our airways have cells that have the cilia. Okay, so it's not the whole thing that's covered, but like, this is where the air is going to be passing by. Okay, and then the cilia is going to be beating and removing trapped particles for you. Like if you breathe in some kind of pollutants or something like that, if you are infected with a cold or something like that, there's a lot of mucus secretion, and you got to get rid of all those mucus, right? And so you will be relying on these cilia to do the job for you. Okay. And where else do you think you will find cilia in the? Sorry, go ahead. Yeah, Micah.
So basically, the cilias are like filters, like just like how the lungs have the little hairs.
Yeah, pretty much, right? Except a filter implies things going through it, like an air filter and trapping things. But these things, it's more like the bristles of a broom, if that makes more sense.
Okay.
Okay. So in the body, there are other places where you will find cilia, which is in the female body, but not the male body. So here is the airway, by the way. Okay, then both male and females have it. This is another location only in females that have the cilia. Anybody want to take a guess at where this might be?
Fallopian tube.
Excellent. That's the fallopian tube. Very good. And what does it do? What does the fallopian tube do?
So fallopian tube can transfer the egg from the ovary to cavity, the uterus cavity.
Excellent. Okay, so in the fallopian tube, the egg is traveling along the fallopian tube from the ovary to the uterus, and sometimes it doesn't know which way to go. So the cilia is going to help guide the egg towards the uterus. Okay, the fallopian tube is also the location where fertilization is going to take place, if at all, and that's where the egg gets fertilized with the sperm. And then once that happens, again, it needs to be guided to the uterus for implantation so that the baby can grow. Flagellum, okay, flagellum is the singular for flagella, okay, and in a cell there is usually just one or at most two flagella. In humans, though, okay, the only place where you're gonna find the flagellum is in the sperm. Okay, so the tail of the sperm helps it swim, okay, and it's a really strong motion because in order for the sperm to find the egg, they have to swim upstream, right, against gravity, right? So the motion is very, very strong and very powerful, and of course it requires quite a bit of energy. So here we have a comparison between the plant cells and the animal cells, and they highlight some features. Some of the things we didn't talk about, obviously you don't have to know, but for example, plant cells, they have the cell wall, they have the central vacuole, they have the chloroplast, right? I don't know why they're circled, actually. On the other hand, for animal cells, we have centrioles, we have lysosome, and then the plants don't have it. Okay. Now sometimes you will see this word centriosome. For us, like it's the same as the centrioles. Okay, like I don't want to make things more complicated than it needs to be. It's already very complicated. I'm sure you agree, right? So if you see centriosome, just think of them as centrioles. Okay, they send out these spindle fibers to help guide the movement of chromosomes. Okay, so just one last question here. Which of the following is made up of two intertwined actin protein strand? I'm just going to relaunch the poll for you. Okay, so nothing mysterious here. If you go back to here and see, it is the microfilament. Okay, this one has two intertwined actin filament. Okay, so that's the answer. Okay, microfilament. That's it for lecture 3. Okay, that's it for lecture 3. I am not going to keep you for too long, but I do want to start a little bit of lecture 4. I just need to draw a picture with you, and then that will be it. Okay, it's gonna take us, I don't know, like 20 minutes or something like that. And then you'll be free to go. You'll be free to go. So let's take out a piece of paper and do that. Okay. Lecture 4 is about transport. Okay, and transport as in like moving things in and out of the cell, and in order to understand transport, we have to understand the structure of the cell membrane. Okay, so that's what we're going to do. Okay, we are going to be talking about the fluid mosaic model. Sorry, I spelled that wrong. Mosaic model of the cell membrane. Right, so here is our cell. with the nucleus. And we are basically going to be taking a slice of the cell membrane and then like zooming in and see what it looks like. Okay. So I recommend doing Skyway. It's not going to be as complicated as the previous one, but we'll draw something here and then we'll take some notes, you know, at the bottom here, right? So plan ahead accordingly. Give me a second, okay? I just need to search up a picture for you to explain what I mean by this. Give me a second, give me a second. Now you gotta understand that the cell membrane is it's three-dimensional, okay? So when we draw it, it's gonna look 2D because that's easier to draw. But you have to keep in mind that it is going to be 3D. And I think the best way to think about it is like this picture. It's refusing to come up, but hold on, it's almost there. Okay, there we go. Oh, there it is. Okay, this is these are called like pool balls, right? Ball pools, I think, like the kids' ball pits. That's what they're called. Okay, so I want you to keep this picture in your mind when we are drawing the cell membrane. Okay, so when I say ball pits, you think of this picture. Okay, it will help you understand what I'm trying to explain to you. Okay, so let's draw it. Okay, let's draw it. We're gonna draw the circle here, and then we're gonna have two tails. Okay, remember this is a phospholipid. Phospholipid. It has a hydrophilic head and two hydrophobic tails. Okay, so we're gonna draw a bunch of these things. This is lecture 4, by the way. Okay, lecture 4. Now some of them might have a kink in the tail. Right? They might have unsaturated fatty acid as opposed to saturated. Okay, it doesn't matter how many you want to put in, just put in a few there. Okay, so like that, and then you know, leave a gap, and then do it again. And then maybe leave one more gap. And then do the same for the bottom, please. Okay, some of them might have a kink. Okay, so this whole thing are basically 2 layers of the phospholipid. So we call it phospholipid bilayer. Phospholipid bilayer. And just on the side here, I want to remind you, this is a hydrophilic head. And then these are hydrophobic tails. So remember, do not draw the next part here. Just watch, okay? Don't draw it. And remember, this is like a 3-dimensional sheet. A 3-dimensional sheet. So like you would have like all the heads, like the ball pits that I was telling you about, right? Like it's like covered the entire cell membrane. You understand that? Okay, we're not gonna draw that. I just need you to visualize that. Okay, covers the entire sheet of the cell membrane. Now these phospholipids can move freely horizontally. Okay, laterally. But rarely flip to the other side. Okay, so just like the balls in the ball pit, they could move, right? They could go across each other. They could flow, freely in front, back, left, right, no problem, right? So if you think about this guy, this phospholipid, it's able to move sideways, laterally, no problem. It just squeezes through between the other ones. But they don't really flip over to the other side. Okay, they don't flip over and go to the other leaf. They can, but it's really, really rare. Is that okay so far?
Yes.
Yeah, thank you. You do have to understand the structure of the cell membrane for your beetroot lab as well. Okay, so make sure you have a clear picture of what's going on here. All right. The phospholipids are not the only things that make up the cell membrane. Every now and then, you will have a protein. That's what the gap is for. Okay, it's for us to like put a protein in there. Okay. Some proteins will span the entire cell membrane. It will go through the entire cell membrane. Others will just be attached either on the inside or on the outside. Okay, so these are membrane protein. If it's just attached on one side, we call them peripheral protein. If it spans through the entire cell membrane, then we call it an integral protein. Okay. Think of a raisin bread. Okay. Some raisins, you can see them on both sides of the bread. Others, you can only see it on one side. So it's like the peripheral versus the integral protein. What do the proteins do? They do 5, 6 different things. Okay. We don't have time to go through it today. I mean, we do have time, but like I want you to do the quiz, right? Instead, so we'll take them up. We'll talk about the functions next time. Okay, but one of the functions is to help maintain the integrity of the cell membrane. Okay, the protein, they are kind of like a cornerstone, if you would, for other phospholipids to build upon around. It's like a foundation, right, for everything else to build around it. Without the integral protein, then the whole cell membrane kind of unravels. Okay. Some protein, they will have sugar molecule attached onto it. Okay, so these little green shapes I'm drawing, that's actually carbohydrates. And the whole thing, the protein portion plus the carbohydrate, this whole thing here, okay, this whole thing, carbohydrate plus the protein, it's called a glycoprotein. Glycoprotein, that's a C. And glycoproteins are important for creating cell identity. Okay. Useful for cell identity. Like, what does that mean? Why does it matter? How does your body know what belongs to your own body and what belongs to the outside? Like if a bacteria goes into your system, how does your body know to attack it and leave your own cells alone? Okay. The short answer to that is, well, they check the cell identity. Right? The kind of glycoproteins, the kind of surface markers that a bacteria has are very different from the ones that you have. So your body is able to distinguish. one versus the other. In addition to glycoprotein, it is also possible to have glycolipids. Okay, so as the name implies, right, it's like glyco means sugar, right? So these are lipids with carbohydrates stuck on them. Okay, so that's the glycoprotein here, and we have a glycolipid right here. Same function, okay? Glycolipid. Also, every now and then, you're going to have a little bit of cholesterol. Okay, not a lot. Yeah. Cholesterol is a steroid, right? Okay, so these are cholesterol. And the function of the cholesterol in the cell membrane is to help regulate membrane fluidity. You do not want a cell membrane that's too fluid. If that's the case, everything's just going to leak out, right? Or come in. We don't want that. On the other hand, we don't want a membrane to be too solid, too rigid either. If that's the case, it lacks flexibility. Nothing can come in, nothing can go out, right? So cholesterol helps regulate the membrane fluidity. Specifically, if you have more cholesterol, then it's going to be more fluid. If you have less cholesterol in the membrane, I'm talking about, okay, then it's going to be less fluid. So whether you want it to be more fluid or less fluid depends on the environment that a cell is in. Okay? If a cell is in a very hot environment, you actually want the membrane to be less fluid. Because if it's very hot, things starts to melt, right? So you don't want it to be too liquidy to begin with. Because the high temperature is going to basically cause it to become even more liquid, right? So we don't want that. If it's in a very hot environment, you want to reduce the fluidity. On the other hand, if it's in a very cold environment, things tends to become more solid, right? So you want your membrane to be more fluid. So it's not gonna like freeze up as easily, right? Okay. Just one final touch and then we're done. This here is the inside of the cell. And on the inside, we have these things. Okay. So these are cytoskeleton. Okay. The proteins needs to be anchored onto something. Otherwise, it's just going to cave in and collapse on itself. We don't want that. Right? We have these cytoskeleton that kind of props it open, helps anchor them down, and help maintain the shape of the cell membrane. Okay. That's it for today. Okay. Please do your quiz, we will continue with this next week. Just print your lecture for next time. If you have any question, feel free to email me. Otherwise, good luck with the quiz. See you guys next time.